So as Patrick said, I'm going to talk about some aspects related to whole genome sequencing. The title of my talk is From Global to Local Perspectives. And I'm going to illustrate my talk with real examples from work that I've been involved with in New Zealand over the last few years. And hopefully we'll be able to take you through this tour in a relatively short period of time. Many technologies involved, and this has been mentioned before, some technologies that have been around for some time, the Illumina technology, others such as the Oxford Nanopore, the MinION, which is about the size of a crunchy bar or whatever. The technology is getting smaller, the data is getting more and more precise, more and more data being made available, and the quality of the information is very much improved. And then in terms of the methods, the downstream approaches, the methods for analyzing the data have become much better as well. And as was mentioned before, whole genome sequencing is just one aspect. There's many other aspects of omics related to this technology which can be applied as well. And Patrick talked about a metagenomic approach for looking at communities of DNA as well. Now this is the slide showing how much the cost has come down over the last decade or so. It's dramatically reduced. So we are able to sequence, depending on whether you want it done quickly or whether you want it done over a longer period of time, the cost of sequencing can vary somewhere between $150 up to $600, $800 per isolate. So certainly the cost is dramatically reduced. Now the kinds of applications that we're seeing, we are applying Campylobacter, as Martin mentioned, in our equivalent of the case control study, the Campylobacter case control study, very similar to the one that's being run in Australia for attributing human illness. Source tracking, I'll talk a bit about later, along the production chain in processing environments, but also this is where food safety meets biosecurity, and this is a key point that I want to make is that biosecurity and food safety are really much linked when we start talking about whole genome sequencing, and hopefully it'll become clear why. And looking at the time and origin of incursions of foodborne pathogens into countries, but also into much more local environments as well. Now, this is an example of an initiative from Mars and IBM together, sequencing the supply chain. You can look at the website and see the details of some of that work that's being done there. This is looking at things like what's normal and abnormal in terms of the microbial population in the supply chain. When can you use this information to detect when things have gone wrong? The sequences generated you can also use for developing diagnostic tests. You can look at antimicrobial resistance, how the pathogens are evolving, and also the relationship between, for example, the genotype and, say, biocide resistance, which can also inform the nature of hurdles that you might want to put in the food chain to inhibit the growth of microorganisms. There are these big international databases with hundreds of thousands of sequences. So the latest one, 2018, we're getting up to 300. I think it may well be over 300,000 sequences that are available through Genome Tracker. You can see that most of them are Salmonella, but we have E. coli, Listeria, Campylobacter, Chronobacter, and Vibrio on those databases as well. And a related initiative, Global Microbial Identifier. These, again, are making sequences available to anybody around the world. And this is great tools, it's great technology, but obviously, as was mentioned this morning, there are some sensitivities around this and the use of that information, and it's important that the wrong conclusions aren't made with reference to these data sets. So I'm going to start globally and talk about some work that we've been involved with, where we've shared data with groups around the world, and here we've been looking at where E. coli 0157, for example, has moved. This is a major pathogen, as was mentioned before, and where has it come from and where has it gone around the world? Now, we use genome sequencing to track the common ancestors of isolates, and we can use where those were found to infer how the pathogen has been transported around the world. And you end up with these kinds of maps. So this is what I'm going to show. The next one is a time series. It's starting actually from the beginning of the 20th century. But what it'll show is the predicted movements of E. coli 0157 around the world. And you can look, fortunately New Zealand is on the map, you can look to see where and when it came into New Zealand. So again, this is available on the web. So you can see movements from Central Europe. It's believed to have originated there. Movement in the 1960s into New Zealand from Australia. And you can see other introductions into Australia and even a suggestion that we've exported 0157 to other countries around the world. So you can look at this in a bit more detail on the website, but it's really an interesting geographical map in space and time of the likely movement. Now, another serogroup of E. coli, which is a toxic group, a sugar toxin producing group, O26. We've done a similar analysis with this. Again, you can only really do this when you compare your sequences with others around the world. And again, it's very interesting, a very similar conclusion. This is probably our most prevalent sugar toxin producing E. coli. And again, it suggested multiple entries of this pathogen into New Zealand in the latter part of the 20th century. Probably about 3 or 4 introductions, most likely coming from Australia or from Central Europe as well. And this is, again, informs things like biosecurity. This is that overlap between biosecurity and food safety, most likely through the introduction and importation of live animals into New Zealand. So that's the big global one. So now we can look at regional transmission, and this is an interesting one looking at Salmonella in Australasia. So this is a piece of work that was done by one of our PhD students which looked at this particular strain of Salmonella typhimurium. It was a wild bird associated strain that started off in Christchurch with a big die-off of wild birds, then transferred over into people and into livestock, and resulted in a 10-year-long epidemic of this particular strain. It was the most dominant strain in New Zealand for 10 years. This paper describes that in detail, but there's an interesting footnote to this. When I was shown some data from the University of Melbourne of strains from Australia, it was quite evident that there was more recent introduction into Australia, but there was this little group here, which seemed to be completely disconnected from the main strain. Cut a long story short, we compared our sequence data with the Australian data, and it fell right in the middle of the New Zealand clade. And when we looked in a bit more detail at the epidemiology of this, we found that this was actually a cluster of cases of people that travelled on a cruise ship from Auckland to Sydney. This is part of a paper that's soon to be published in September, but again, it's a really good example of exchange information of how you can infer, in this case, inter-country movements of key foodborne pathogens. Another example, now we're focusing down a bit more closely to home, and this is a focus really mainly on North Island. In 2014, we had the emergence of a new strain of Campylobacter, which was resistant to 2 classes of antibiotics. And this was a real step change for us, because up until this point, we'd had very low levels of antimicrobial resistance, both in human cases, but also in animal reservoirs as well, including in chicken. This strain came in, and the unusual thing was it was very quickly found in all the different poultry supplies in North Island, not in South Island, but ultimately found in 4 the major poultry suppliers, and also in breeder flocks as well. So it was associated with outbreaks. There was a chicken liver outbreak here in Wellington, which was associated with this particular strain. Now, how we've used genome sequencing in this case is to look at how this pathogen spread so rapidly around the poultry supply in North Island. So we sequenced those isolates, then we looked to see how the genetic relatedness of that then compares with things like the road distance or the geographical separation between farms, whether they share a feed company, whether they share personnel, all those kinds of things. And again, to cut a long story short, there was really strong evidence of within company spread, so associated with parent company, but also geographical spread, and very strongly associated with the road distance between farms, and also some other aspects of the network as well. So we can use the data to inform how it spread, and use that information then to devise better biosecurity or control strategies to prevent spread between farms. Again, locally, just thinking about outbreaks, so we have used whole genome sequencing in a number of outbreak situations as well. This is the outbreak of Yersinia pseudotuberculosis in 2014. It was used more retrospectively. I think if we'd had used it more in real time, then maybe the source of that outbreak may have been determined more rapidly. This is one of the largest Yersinia outbreaks anywhere in the world, over 200 cases, and many of those hospitalized. Although fresh produce was implicated, the precise source was never really identified. And I think if we had better databases, better sequencing, more rapid sequencing, we may have got ahead of the curve, as Martin was talking about, a bit quicker with this one. Another outbreak, not foodborne, but waterborne, was the application of whole genome sequencing in the Havelock North waterborne outbreak. Again, probably the biggest outbreak of waterborne Campylobacter anywhere at any time that we're aware of. Over 5,000 cases in a town of just 15,000 people. And again, whole genome sequencing was one of the tools that was used to identify that the strains that were found in sheep in the reticulated water supply and in the people were virtually identical. So that was really good evidence that it was the sheep contaminating the water that then resulted in the infection in people. So that's another example of an outbreak, not a foodborne one, but of a foodborne pathogen. The last one is nothing to do with food, but again is an illustration of how we're using genome sequencing in real time, in this case, to track and trace the source of an outbreak. So we're working with MPI, in particular with the Mycoplasma bovis sequences. We are using this information to do two key things. One is to determine what the likely date of origin, so when this pathogen came into New Zealand. And by using this ancestral dating methods, it can really help to tie things down, and can also inform whether or not it's feasible to go for an eradication strategy. So this is really important for determining and helping decision-making at government level as well. And we also use these, combined with the information on when herds were likely to have got infected, to track and trace the transmission between individual infected premises over time. So I've given 3 examples of its application in outbreak situations, but it's foodborne, waterborne, but also other outbreaks can be used as well. This is an example of a visualization tool. These are some of the tools that are really coming on stream now. This is an online tool where you can actually upload information, you can get the phylogenetic sequence, you can actually map them, these two are interactive, so you can click on here and it will show where the sequences are on the map as well. This is a tool called MicroReact, a very useful tool. Local transmission studies. So what we're doing is working with a number of organizations, companies in particular, and we're looking at contamination of food processing environments and how we can use whole genome sequencing to determine entry points, vectors, transmission chains, spatial spread around production environments. We're using techniques which have previously been applied to transmission of antimicrobial resistant strains, such as MRSA, in hospitals to see if we can track and trace the origins of these. There's a lot of very interesting tools and techniques that we're developing in the Food Safety Centre at the moment related to this, and we're hoping to extend this technology considerably over the next few years. So we're now down to the very local, and this is about as local as it can get. This is actually a poor patient who had Campylobacter infection for 10 years. So they repeatedly showed up at our Sentinel surveillance site with Campylobacter jejuni, and it was one particular strain. So we used genome sequencing of isolates from this patient over a 10-year period to ask the question, is that person picking up repeated infection from the environment, or have they got an infection inside which is located in them, which is currently constantly developing and evolving over time? Again, to cut a long story short, what this showed was that this patient was probably chronically infected with an internal Campylobacter. And the reason why we think that is because the strains are very similar over time. And when we looked at multiple strains at the end, it was suggesting that antimicrobial resistance had developed when that patient was giving antibiotics. So when that patient was given an antibiotic, the Campylobacter developed resistance. And there was evidence of what we call selective sweeps in that population. So the population declined dramatically over time whenever that patient was given antibiotics. So this is an example of using whole genome sequencing at the highly local level. So, to summarize, hopefully, it's a quick whistle-stop tour through, but just using examples from New Zealand and from studies that we've been doing here. We're using whole genomic sequencing extensively and increasingly routinely. We literally have thousands of whole genome sequences that we've been using over time, working with many of the partners within the Food Safety Centre as well who have whole genome sequencing capabilities. So working with ESR, also working with AgResearch and with Massey University, using the technology increasingly routinely for different applications. We can look at global transmission, look at that from a biosecurity as well as a food safety perspective. Local transmission, food processing environments, outbreak investigation, attributing source, and looking at things like antimicrobial resistance. And the databases, the more we go on, we're developing better tools for visualization, for analysis of the data, much more accessible tools, and improving our ability to make good decisions in the food chain. So I'd just like to leave it there, and a huge number of people involved in all of the work that I described there. Most of it's published, so if you do want to get that further information, you can get that readily available.